So for this section, we're going to talk about toxins. We're going to describe the 9 basic cellular targets for the bacterial toxins. We'll talk about the mode of action, specifically for staphylococcal alpha toxin, cholera toxin, diphtheria toxin, Shiga toxin, and anthrax toxin. And then we'll talk about the difference in endotoxins and exotoxins. So here's the case history, the telltale cough from your textbook. Will, a formerly rambunctious 5-year-old from San Francisco, was taken to the emergency department after having a severe cough for 2 weeks. His illness started as a runny nose, dry cough, and low-grade fever. But within days, the coughing would come in violent fits lasting up to 1 minute, called paroxysm. The nurse practitioner attending Will observed one of these fits. Unable to breathe between coughs, the boy became cyanotic, or he turned blue. When the staccato of the coughs finally ended, Will gasped, desperate for breath. The air rushing to fill his lungs made an ear-piercing whooping sound. This telltale whoop led the nurse practitioners to suspect whooping cough, a disease highly contagious in its early stages. The nurse practitioner, now wearing a mask, took the nasopharyngeal swab sample from bacteriological culture and prescribed the antibiotic azithromycin, which is a member of the macrolide class of antibiotics that inhibit protein synthesis. Because the organism takes so long to grow in the laboratory, 5 to 12 days, and because the organism is hard to find late in the disease, a blood sample is also drawn to test for anti-B pertussis immunoglobulin A antibody. By the next day, the serum test was positive for B pertussis antibodies. The pathogen eventually also grew in the culture. Despite extensive efforts at vaccination, more than 41,000 cases of whooping cough were reported in the United States in 2012, 33,000 in 2014, and the number has continued to increase through the years. So let's first talk about the actions of the bacterial exotoxins. And basically we're talking about superantigens that trigger pretty severe systemic responses. Staph aureus has a TSST-1 toxin and an exfoliative toxin, and Bacillus anthracis has a lethal toxin. But in general, here are the different targets that you can see that are impacted by the bacterial exotoxins. For example, the plasma membrane disruption can be affected by Staph aureus A toxin, Staph aureus leukocidin, perfringolysin, E. coli A toxin, Clostridium perfringens enterotoxin, Vibrio parahemolyticus hemolysin. There's also a group that affect the cytoskeleton, like the C. botulinum C2 toxin, C. perfringens iota toxin, and Vibrio cholerae RTX. Another target, protein synthesis disruption, the diphtheria toxin, Pseudomonas aeruginosa exotoxin A, and the Shiga toxin. Another target, cell cycle disruption, the cytolethal distending toxins as well as the Pasteurella multicida toxin. Another target, signal transduction disruption, E. coli STLT, Clostridial cytotoxin, the cholera toxin, pertussis toxin, Bordetella DNT, anthrax toxin, and superantigen. The cell-to-cell adhesion disruption by staphylococcal exfoliative toxin and B. fragilis toxin. Vesicular trafficking by H. pylori, VAC-A, and then the aerolysin. And then finally, exocytosis as a potential target. C. botulinum neurotoxins and tetanus toxins. Let's look at some of the details of some of these types of toxins. Specifically, a 2-subunit AB toxin includes a toxic A subunit and a B subunit that binds the host cell receptor. Many of the B subunits are complexes of 5 units arranged as a ring. The ADP ribosyltransferase inactivates or alters target proteins, and that is shown here in the graphic. Here we see the Staphylococcus alpha toxin. When this toxin actually affects the blood cells, we see something that's called beta hemolysis. This is a relatively clear breakdown of the blood cells. Cholera and E. coli have a labile toxin, which means it can be broken down. The opposite would be a stable toxin. In this case, the cells in the villus will absorb the glucose, the sodium and chloride ions, other ions, and water from the intestinal contents and pass them into the blood. The crypt cells will remove the electrolytes and water from the blood. Anions such as chloride move into the intestine by the CFTR channel. Anthrax toxin. It has two A subunits called edema factor and lethal factor. The B subunit is called protective antigen. Edema factor causes fluid loss to the extracellular spaces, very similar to the Bordetella bacteria. The lethal factor destroys regulatory cascades and cripples the immune response. Here in the graphic, you can see the protective antigen subunit is made as a single peptide. The protective antigen then binds to the host cell surface. 7-PA fragments autoassemble in the membrane to form a pore. The other two components of the anthrax toxin, EF and LF, bind to the ring and they're carried into the cell by endocytosis. And then EF and LF are expelled through the PA pore into the cytoplasm. Endotoxins. These are called endotoxins because they're actually part of the membrane itself of a gram-negative cell wall. And this includes the lipopolysaccharide in the cell wall. So, in essence, the bacteria itself is a toxin. Fever, activation of clotting factors, activation of complement, vasodilation, shock, death, all these can result when the endotoxin is released into the blood. This next section, 18.4, talks about secretion of virulence proteins. We'll differentiate between type 2, 3, and 4 secretion systems that are used to export the exotoxin and effector proteins. And we'll talk about the evolutionary relationship between the secretion systems and the other microbial structures. This chart shows you the different secretion systems for the bacterial toxins. Secretion type 1 has a SEC-A dependent component, so one effector per system. And we see this with E. coli alpha hemolysins, Bordetella pertussis adenylate cyclase. Secretion type 2 is a SEC-A dependent program similar to type 4 pili, Pseudomonas aeruginosa exotoxin A, elastase, and cholera toxin. Secretion type 3 is a SEC-A independent. There's also a syringe type in this, which we'll show you in one of the next slides, related to Flagella. It secretes multiple effectors from the bacterial cytoplasm directly into the target host cell cytoplasm. Yersinia YAP proteins are examples, Salmonella CYP proteins, enteropathogenic E. coli, ESPA proteins, and TRA. Secretion type 4. It's related to conjugational DNA transfers. Secretes multiple effectors. The Bordetella pertussis toxin and Helicobacter CAG-A are examples. Secretion type 5. It's an autotransporter, SEC-A dependent to periplasm, self-transport through the outer membrane, and we have one effector per system. Examples, the gonococcal and Haemophilus influenza IgA proteases. And then finally, secretion type 6 is related to phage tails, a single effector. Burkholderia and Vibrio cholerae, VGRG, are examples. So let's look at type 2 in this graphic. This is called a piston delivery system. This shows you the outer wall and the general secretion pathway, which actually comes through special pores from the periplasm to the outer membrane and to the lipopolysaccharide. The type 3 secretion system is a syringe type delivery, and it's called that because it is very much like a syringe, like a needle. Here you can see that it actually extends outward from the outer membrane, and then it actually penetrates into the membrane host cytoplasm. Type 4 is somewhat similar, except it's a tube delivery system. Section 18.5, we're going to talk about the extracellular pathogens, or differentiate them at least from the facultative and obligate intracellular pathogens. We'll discuss the three main ways intracellular pathogens avoid intracellular destruction. We'll describe the various strategies the pathogens use to avoid the immune system, and we'll explain why and how intracellular pathogens distinguish between intracellular and extracellular existence. So first, the intracellular pathogens. They seek refuge inside a host cell. They're not detected by many components of the immune system when they're inside the host cell, and so this is one way they can evade the immune system. Facultative intracellular pathogens are one example. They can invade the host cell, but they can also survive outside the host cell. And examples are the Salmonella species, Shigella, and Listeria. Obligate intracellular pathogens invade and reproduce inside the host cell only, so they cannot live outside of their host. Examples are the Rickettsias, Coxiella, and Bartonella. This shows an example of how the intracellular pathogens operate. So a bacterial pathogen attaches to the host cell membrane. The pathogen induces phagocytosis, and once it's inside, it combines with another vesicle to become a phagosome. The pathogen has one of three fates depending on the pathogen. So fate 1 down here, the pathogen such as Coxiella burnettii allows phagosome-lysosome fusion and differentiates into a form able to replicate in the phagosome, resulting in inclusion bodies. Fate 2a, pathogens such as Salmonella can remain inside a phagosome and prevent fusion with the lysosome. Fate 2b, the Salmonella typhi will remain in the phagosome, which moves to the host membrane and expels the Salmonella into extracellular spaces. 2c, from there the bacterium can be engulfed by the macrophage and survive within the phagosome. Fate 2d, the macrophage can travel to regional lymph nodes and disseminate the organism through the circulatory system. And then fate 3, the pathogens such as Shigella and Listeria break out of the phagosome and then move throughout the cytoplasm into the adjacent cells by forming actin tails. Extracellular pathogens do have a way to avoid the immune system. If they form a capsule, they can actually evade phagocytosis, as you very well know from both earlier lectures in laboratory. They can have self-surface proteins that are components of the cell wall that prevent detection. They can bind to the Fc region on the antibody. They can alter the antigens to avoid antibody binding. And then we have quorum sensing that's used to communicate with the other pathogens about the population size. This graphic shows Salmonella and shows a model of the bacterial pathogenesis. As you can see here, Salmonella attaches and has a type 3 secretion system. It produces effector proteins that then cause gene expression of certain chemicals such as cytokines, which will then have other effects on your system, like in your intestines. Section 18.6 talks about viral pathogenesis. We'll discuss the antigenic variation in the rhinovirus and influenza viruses, explain how the human immunodeficiency virus targets the T cells. We'll describe the roles of NEF and TAT proteins in HIV pathogenesis. We'll outline how human papillomaviruses cause unregulated cell division. We'll explain the latency of herpes virus and the role of microRNAs in helping the virus reactivate. So, for immune avoidance by viruses, there is antigenic variation, so there's 100 known serotypes of rhinovirus. Each virus has a unique capsid protein. Antibodies to one capsid protein are not effective on another. There's also antigenic shift. Two strains of influenza virus can infect the same cell, and the genomes can get mixed. It makes a dramatically different virus in that case. In antigenic drift, we have random mutations that occur within the cell that a virus infects, and this creates small changes in the virus proteins. One specific example that we'll talk about in viral pathogenesis is human immunodeficiency virus. HIV binds to the CD4 receptor on the T helper cells, as well as the chemokine receptor CCR-5. HIV inhibits apoptosis in T cells and allows the cell to survive longer to make more virus. Look at this table 18.4. This shows the HIV proteins that affect pathogenesis.